with our next seminar speaker. We're going to hear from Evelin, and she's going to be introduced by Graham Sazama.
I'll get the woos. All right. I am very pleased to also introduce this afternoon, Evelin Campos Salazar. So I am technically Evelin's advisor, even though I'm not allowed to advise biochemistry students. But Evelin came to me in a time when there weren't too many people around to advise biochemistry students, and so with that, I got to know Evelin a lot better, and pretty excited to kind of hear her story and where she wants to be. So Evelin came to Lawrence as a Schuler Scholar out of the Chicago area. She comes from Highland Park, Illinois, and has since really made a name for herself in the laboratory. I think she's got a bit of a reputation as really rocking it in the lab, and worked with Stefan Debert in his lab for several terms and at least one summer, right? One full summer? Yeah. Of course, everyone dealing with the COVID year, so nobody worked then. But in any case, through that, I think Evelin really fell in love with the idea of being a pharmaceutical scientist, and so was looking to take a gap year, get into the industry in some ways, and then eventually find her way to grad school in the same thing. So we'll get to hear about her first forays into that field right now. So without further ado, here's Evelin.
Can everybody hear me okay? Awesome. All right, well, thank you, Graham, for the introduction, and thank you for everyone coming and being here today. As Graham said, my name is Evelin, and today I'll be talking about nitrogen heterocycles and medicinal chemistry, and a little bit about my summer research as I made various compounds using nitrogen heterocycle. In fact, the compound on the screen is actually the compound that I spent the entire summer trying to make. And to just give a little bit of a rundown on what I'm going to discuss, first off, I'll be starting off with some background information on heterocycles and nitrogen heterocycles and how their diverse biological properties and chemical properties make them significant in medicinal chemistry and drug development. Specifically focus on a specific type of nitrogen heterocycle called triazoles and their relevancy in medicinal chemistry as well. Which brings me to my summer research. Sorry about that. About my summer research, where I explore various triazole compounds using a variety of methods, and which all relates to the treatment of schistosomiasis, which is a parasitic disease, and discuss the connectivity to an antimalarial drug. And then I'll just dive into some lab data and findings of the triazole compounds that I attempted to make over the summer, and then dive into some other research, a specific study that explores various triazole compounds and their potent antiparasitic activity against schistosomiasis, and I'll just wrap up everything at the end. So to start off with some background information on heterocycles, heterocycles are carboxylic compounds where the carbons are in a ring structure, as you can see with the blurred picture in the background, where one or multiple carbons are replaced by a hetero atom. So in terms of a nitrogen heterocyclic compound, these compounds or these cyclic compounds contain one or more nitrogen atoms. These type of heterocycles are important as they are widely used in pharmaceutical sciences. They come in all different shapes and sizes, so they will consist of a 4-member ring, a 5-member ring, and so on and so forth. They're considered to be important building blocks as the nitrogen atoms allow for diverse bonding, and they also contribute to the aromaticity of a heterocyclic compound, which just plays a role in stabilizing the entire compound and changing the reactivity of the compound as well. These heterocycles also tend to have great solubility properties, which makes them more effective in drug development as we want drugs that are easily absorbed. So now allow me to introduce to a special nitrogen heterocycle called triazoles. These are 5-member rings containing 3 nitrogen atoms, and they're known for their non-covalent binding. So this just means that it allows for variation of interactions between or within a molecule. As triazoles consist of more than one nitrogen, the opportunities of hydrogen bonding, there are more opportunities for hydrogen bonding among other articles. So as you can see in the picture, there's a triazole ring right there, and you can see this nitrogen bonding with this oxygen from basically this is just like a amino acid, the side chain of an amino acid residue. And so this is just a picture of a protein ligand binding interaction. And so, yeah. And so that's what I mean by hydrogen bonding. So what makes these triazole compounds so special? What makes them very special is the ability of these nitrogen atoms to form hydrogen bonds with a variety of biological targets. And these are just a few out of the many biological properties of triazole. Most of the triazole compounds are used in fungicides and for antiviral drugs, but they carry various other biological properties that make them useful and significant for considering them in drug development, especially for diseases with limited treatment. For instance, schistosomiosis. Schistosomiosis is a significant disease being explored in medicinal chemistry. Schistosomiosis is a disease caused by a parasitic worm contracted through fresh water. So the worm penetrates your skin when you're in the water and travels through the bloodstream and goes into your reproductive areas, which can cause long-term irreversible health implications like bladder cancer, fibrosis, and other reproductive issues. Schistosomiosis is widely spread, mostly in poor communities, infecting around 221 million people. Yet the bigger issue lies within the limited treatment available for schistosomiosis. One of the current treatment available for schistosomiosis involves praziquantel, which is an oral treatment that tends to be effective within a few hours and only taken for a couple of days. It works by causing the severe muscle spasm and muscle paralysis in the worms, which all sounds pretty great. It's a very effective treatment, but again, with limited treatment comes limited options, which makes it very hard to accommodate to every individual's health needs. And then the larger issue is that eventually we will face drug resistance, which will happen where the drug will become less effective over time. So how do we develop more drugs for schistosomiosis or against schistosomiosis, and how do we approach this problem of drug resistance? Well, we start off what we do know, and we start off with the drugs that already exist. So here we have an antimalarial drug that I will refer as a lead compound that was already explored and found to carry potential antiparasitic activity against schistosomiosis. Now that we have an idea in where to start, now that we have an idea of where we want to start, we want to ask ourselves how we can improve our lead compound and achieve the effect that we want, how to better the activity specifically against schistosomiosis. So it starts off with just making these small chemical changes, like playing around with different heteroatoms, acidicity, basicity, explore around it and see what else there is to it. But also make these changes with compounds that we know are useful and carry diverse potential and relevant properties. In this case, we have an antimalarial compound used for schistosomiosis. There's various areas where we can make these small chemical modifications, but the research I did focused on this heterocyclic linker, where the furan is at right now, the furan referring to the 5-member ring with the oxygen. And so we want to replace it with other heterocycles that may contribute to potent antiparasitic activity. So why not use a nitrogen heterocycle, specifically a triazole? As I discussed earlier, we start off with what we do know, and we know that triazoles carry diverse biological and chemical properties that could be very beneficial when considering them for treatment for schistosomiosis. And this is more like a trial and error process in the sense that we want to use compounds that we know may be beneficial in drug development. And theoretically, it sounds like a great idea. It makes sense. but we don't really know in reality how they're going to react when we put them to the test. So now that we have an idea of what we want to make, we have to figure out how to do so. And that's where my summer research comes in, where my targeted compound is the one on the screen. And so it's just coming up with the synthesis plan and how we're going to incorporate that triazole compound in our lead compound. So how to make the 1,2,3 triazole in the first place. And the 1,2,3 triazole just refers to the positions of the nitrogens on the ring. And this is what most of my summer research consisted of, exploring various methods and alternative ways in making this triazole compound, specifically a procedure plan informing this 1,2,3 triazole ring. And then we want to check and test if we ended up making the compound in the sample. So after extensive research and what past literatures have done, I decided to try out this route of synthesis, where we start off with the starting material 4-chloroaniline, going into this azide compound, and then later on forming the 1,2,3 triazole ring. The first part of the synthesis, I utilized two different methods. One of the methods didn't really work out, so I ended up resorting to a different method, which is why I used two different ones. And then for the second part of the synthesis, it consisted of a basic click reaction. So the first method I performed for this first part of the synthesis was using an amber-Lisk ion exchange resin to form the desired disonium intermediate, and then using sodium azide to eventually get to the azide compound. The intermediate allows for an easy access to get this azide compound, as it is very electron rich, which means that these bonds can be easily manipulated to form other bonds. So why use this resin in the first place? Well, on paper, it all seemed like a pretty good idea. It all seemed ideal. It produced high yields. It was a safer procedure. And it was a useful catalyst in selective removal of any excess starting material or reagents and any other unnecessary byproducts that we want to avoid producing throughout our procedure. But it didn't end up working out. And it's just, I just ran into a lot of procedure complications when it came to actually doing the experiment, and we didn't end up getting the high amounts of the azide as we were hoping for. So we ended up resorting to a more traditional method that led to be more successful, using hydrochloric acid that we didn't use in the first place because we wanted to try alternative ways first to see if those alternative ways were better. And we wanted to avoid using more corrosive chemicals, but this method ended up being more successful. It still led us to this disonium intermediate that would eventually lead us to the azide compound when we add in that sodium azide in there. So how do we confirm the presence of the azide? How do I know if I actually made the azide compound after that first step of the synthesis? Where there's two ways to know that. GCMS, gas chromatography, mass spectroscopy, and then the nuclear magnetic resonance spectroscopy, also known as NMR. I mostly use the GCMS to confirm the presence of the azide. And the GCMS basically separates substances in a chemical mixture and then identifies those substances through their molecular weight. And NMR measures the magnetic force of a nuclei, which helps identify the structure of a molecule. As for the triazole part, I only use NMR as the GCMS wouldn't recognize it. So we couldn't know for sure only through the GCMS. So we had to rely on the NMR to really identify whether I made that complex 1,2,3 triazole compound. So this graph shows the GCMS of that first method using the resin. As you can see, there are traces of the azide. However, there's also the presence of the 4-chloroaniline and other byproducts that we just don't want. And if we compare this to the second method of using the hydrochloric acid, it just gave us cleaner results, where we just wanted mostly the azide and very, very low amounts of any other byproducts or the starting material, which will be very beneficial in going on with the second part of the synthesis, as it'll be easy to purify at the end. So for the second part of the synthesis, it involved a click reaction where it consisted of reagents of propargyl alcohol, copper sulfate, and sodium ascorbate. And so this is called a click reaction. A click reaction is also known as click chemistry. It's a chemical reaction that occurs between reagents to solely react with each other. And they are known to produce high yields. They're also known to be like fast and simple to perform and easy to remove any solvents that are used throughout the process. So the experimental procedure consisted of adding all these reagents together with the prepared azide compound from the last synthesis and a solution of solvents. And I let that stir for about 36 hours and removed the solvent and then tested whether I was successful in making the 1,2,3-triazole compound. So how this chemically works is basically the triple bond on the propargyl alcohol is going to open up and react and form or react with one of the nitrogens on here on the azide, and then basically a bunch of electrons are just going to be exchanged, eventually leading to the formation of the triazole ring. And then we end up also with the attached alcohol group. And we want that alcohol group at the end as well, because that's going to act as our reactive group, where later on we can add more onto the compound and vary its length and size. So this graph is a Proton Nmr graph of the sample of my attempted 1,2,3-triazole compound. So we expected to see peaks at 7.9, 7.7, 7.5, and 4.8. And these peaks were based off other literatures and other research where they observed their peaks. And so I knew roughly that this is where I should be expecting my peaks to be at. These peaks represent the ratios of hydrogen atoms and what type of hydrogen atoms they are referring to. The S and the D are referring to singlet and doublets, which is more specific to how those hydrogen atoms are being shared based on their environment. So for example, each letter corresponds to the specific hydrogen that the peak is referring to. So for B, those two hydrogens are very similar to each other as they are surrounded by the same environment. So that's why we expected to see a doublet at that specific peak. So based on this graph, we were able to match up the hydrogens from the triazole compound with the peaks confirming the presence of the triazole. The alcohol group especially, the alcohol group was a very clear peak from all the past literatures, and the azide as well, corresponding to letters D and E. So to conclude everything and then just the NMR, I was successful in making the compound at the end of the day as those peaks corresponded to the 1,2,3-triazole, the alcohol group, and the azide. And so these are just some pictures of the actual experimental part of the lab that I did over the summer, where that first picture is basically the using the hydrochloric acid to form the azide compound, where I put it in an ice bath. And then the far right picture is the click reaction. The middle picture there is just an aqueous workup. And the bottom picture is the azide sample. That's what I ended up with, and that's what I tested on the GCMS. And then that's just my NMR scan and Stefan explaining to me how that all works, because it's still confusing. So going back to the steps, just to summarize, we started off what we do know. We make these small chemical changes. We come up with the synthesis plan and how we're going to make those triazole compounds. And now we need to actually test to see if they actually carry any potential antipyric activity for towards the biologic targets for systemiosis. So this is where the biology part comes in and understanding how the triazole compound interacts with the biological target. Personally, I did not get that far within my research over the summer, but for the rest of the presentation, I will be talking about a study that does further explain the triazole derivative they explore and the biological target and the analysis of it. So this article is by Dr. Dimitri, where they would talk about all these triazole-based derivatives they worked on and using them as potential inhibitors of HDAC. So getting into the study, these are the derivative compounds on the screen that they explored in literature before. So they didn't come up with these compounds. These compounds were already explored in past literatures various times. But in this specific study, they are mainly focusing on synthesizing these compounds using alternative methods and routes involving fewer steps, than the ones they used in past literatures. The alternative routes that they perform involve similar procedural methods to what I did over the summer, and they ended up with compounds like the ones highlighted in blue. And the key part of this compound is this like hydroxyl group, which is this group on the end. And as it's the reactive part of the compound, which I would explain how later on in the presentation. So connecting back to testing the antiparasitic properties of these triazole derivatives, in this article, they focus on targeting HDAC and using inhibitors of HDAC as a possible drug development treatment for systemiasis. So what are HDACs? Well, they're histone deacetylase, which are specific enzymes that work to catalyze acetyl groups from a specific amino acid on histones. And it makes the histones wrap tighter around the DNA. And this plays a crucial role in just regulating gene expression, like inflammatory genes in humans. And then later on, and further in the presentation, I will talk about SM-HDAC and HS-HDAC. And basically, they just differ the enzymes, and the SM refers to the enzyme in the parasite, and then HS refers to the enzyme in humans, as they want to observe the activity that the triazole derivative have with both the enzymes. So this specific enzyme, HDAC8, was found in all of the parasite life cycle, which will make it a crucial target and a possible route of treatment for systemiasis. And what inhibition would do the downregulation of HDAC enzymes would decrease the parasite's capacity to survive, which makes it a very crucial, like a key feature in targeting HDACs in the parasite. This table shows the inhibition activity of triazole derivatives that they created. So they found that the triazole-based compounds 2B, 2E, 2F, and 2G were the most potent inhibitors of HDAC in the parasite. They basically determined this based on the values over 50% of inhibition against the parasite's HDAC enzyme. Out of all of them, they determined that 2B compound, the triazole-based 2B compound, as the most potent inhibitor against HDAC, which was further explored in the study and what I will be focusing on in this presentation as well. So here we have a crystal structure on how the inhibition looks like interacting with the enzyme. So in this first figure, we can see the complex of the HDAC enzyme with the triazole derivative compound. And then the second figure is a more close-up look of the actual binding of the triazole derivative compound within the active site of the enzyme. And then you can see here by the white dashed lines that the side chains of the HDAC enzyme of the parasite is binding with the reactive groups, carbon atoms of the triazole compound inhibitor. So that's the hydroxyl group that I was referring to. That's the reactive group. That's what's interacting with these side chains of the enzymes. So some key findings from this study, it provided some insight in the interactions between the triazole ring and both the enzymes involved in humans and in the parasite. And that just all contributes to specific selectivity towards to both specific enzymes. For example, due to the lack of binding between the triazole ring and the amino acid residues in human HDAC enzyme, that caused lower percentages of lower inhibitory activity, which is what we do want. We want to reduce the inhibitory activity towards human enzyme, and we want to increase the inhibitory activity against the enzymes in the parasite. And then, so all of this is just revealing interactions of the triazole ring with the active site, which this all prove that triazoles to be promising compounds to be considered as potent SM-HDAC inhibitors, so enzymes in the parasite. So what still needs to be done is just improving selectivity of this triazole-based compounds derivatives that they came up with, selectivity towards the parasite, and decreasing the inhibition in human enzymes, because we want to increase the selectivity towards the enzymes of the parasites just because we want to decrease their capacity to survive. To wrap everything up, as we went through all these steps, they all relate to how drug development work in medicinal chemistry, how we approach problems like drug resistance, starting off with what we do know and narrowing it down to specifics like triazoles, and then applying it to challenges that we are trying to face. However, there's still a lot more of learning to do, as not all of these concepts are completely understood. Like I said, it's like a trial and error process, as we start off with an idea, we run with it, an idea starting like nitrogen heterocycles, as we know that they carry all these diverse biological and chemical properties. And then we run with it with the hopes of achieving better results within each step that we take. The important thing to understand is that it's not a simple process, which is something that I learned over the summer. There's a lot of back and forth that I had to do over the summer, which is why I didn't get to that last part in testing the biological target and seeing and observing its antiparasitic activity. But although nitrogen heterocycles are great, the application of them into drug development is much more complex, as medicinal chemistry, as I mentioned, is all a learning process. So I would like to acknowledge my advisor, Dr. Stefan Debert, just for giving me the opportunity to do the summer research and continuing this journey with me throughout the term as well. And then I would like to thank Dr. Graham Sasma for all of his support and for the lovely introduction, and Dr. Deanna Donahue, because she played a huge role in making this presentation, just preparing me for today, as well as Professor Rachel Daughtry, who also helped me prepare for today, and for DMARC for, because this wouldn't be possible without you and actually doing the experimental process, and for replacing all the broken glassware that I caused, and then the Claire Booth Lou Scholarship for just allowing me the opportunity to do the summer research, and then Ezra, Emily, Carmen, Matthew, and Chris, who were all part of the summer research program as well, who played a role in just the research that I did over the summer, and all of my colleagues in Chem 680 as they gave me feedback for this presentation today. And that'll be it.
All right, the floor is open for questions. Yes, over there, Adriana.
On slide 26, you talked about the side chain interactions of the triazole compound. Is there any interactions directly with the triazole ring in the active site?
Okay. It says slide 26. Yeah, so yeah, here I did talk about mostly about the interaction of the hydroxyl group with the side chains of the active site. And so to actually, and you're asking about the actual triazole ring, which is this part of the ring over here, and how that incorporates to all of the rest of the active site. So that part of the article was very complex because they were talking about how the orientation of the ring contributed to just stabilizing the entire compound and it allowed for some electron stacking for this hydroxyl group with the active site. They didn't really go much in depth about it. They just said it that way. But yeah, so I would just say that the triazole ring just kind of contributed to the reactivity of the hydroxyl group with the active site.
Other questions from the audience? Kelly?
This is probably a little out of the scope, so don't tell me to stop. But so this is showing your cystis HDAC here.
No, so this yes. So this is referring to the cystomyosis HDAC.
Did they talk at all in this paper about how the human one would be different? Like how possible differences in this protein structure?
If I'm being completely honest, I mostly focus on the parasite structure.
Mm-hmm.
The human one, they mostly just talked about how there wasn't much reactivity with there was low inhibition activity towards the human enzyme active site, which was just kind of the key finding that they mostly talked about, but they didn't really show any crystal structures like these. They mostly just focused on the parasite.
Yeah, cool.
Yeah.
Any other questions for Evelin? If no, let's thank our speaker again. Great work today. We have no seminars next week in honor of reading period. Join us again week 7, Tuesday, for Carmen's talk.